So my name's Sadie. I'm a Ph.D. candidate at Washington University in St. Louis. So I'm a senior grad student in Samantha Morris's lab, and we work on using single-cell technologies and single-cell lineage tracing to understand mechanisms of development and disease, typically, and what I'm going to present today, in the context of cellular reprogramming. So how cells make decisions as they go through transitions catalyzed by transcription factor reprogramming.
Yeah, I'm Zohar. I'm Associate Director of Emerging Technology at Ultima Genomics, and today I will talk with you about single-cell genomics at scale and how we do it in Ultima Genomics on the UG100. And I will let Sadie talk first about our beautiful project.
Great, thanks for the introduction and thanks for the opportunity to be here today. So I'm going to be presenting my work titled High Resolution Single Cell Lineage Tracing at Scale Reveals Reprogramming Dynamics. And this is a very exciting project that we actually just worked on at the end of last year with Fluent Biosciences and Ultima Genomics in order to accomplish single cell lineage tracing at much larger scale than we've previously done before in the lab. So cell reprogramming transforms cell identity from common cell types into cell types that have major clinical relevance. However, these cell type conversion protocols generally lack efficiency and don't always convert cells to the directed final cell fate. And of the cells that do reprogram, we can often identify unintended off-target reprogrammed cell fates and inexact matches to our desired targeted cell fate. So the cells that we generate from these protocols do not fully reflect their in vivo correlate, even if we are able to typically transplant them and still get a functional cell type at the end. So to better resolve how cell reprogramming succeeds and fails, we have to understand cellular characteristics that contribute to these outcomes. And broadly, these characteristics can be broken down into two categories: cell intrinsic effects and cell extrinsic, or these community effects. So cell intrinsic effects are changes that occur within a single cell as it grows and divides, and that can lead to successful cell fate conversion. Cell extrinsic effects refer to the outside pressures on a cell, such as competition within cell culture, that can impact cell fate conversion as well. And by leveraging single cell lineage tracing methods here in the center, we can actually tease apart these characteristics to better understand their roles in cancer, development, and regeneration. Specifically, if we assess differences between clones, groups of cells all derived from one progenitor cell, we can infer how the initial cell state of the progenitor cell and the external effects on the population affect cell fate conversion. And then if we go deeper and assess relationships between individual cells within a single clone, so exactly how these individual cells are related to each other, we can infer how changes in cell state over multiple cell divisions can affect cell fate conversion. So to measure cell lineage at single cell resolution, we employ two major approaches as a field. The first generates random barcodes via DNA synthesis and tags cells with these barcodes. Cell tagging uses this approach and lentiviral integration to insert this lineage barcode into the cell's genome. The barcode is inherited by all progeny from that initial cell, and this confers a clone label to define the cell's progeny, and that information can be recaptured from any mRNA recapturing method. Second, we can create random and unique edits within a cell's genome that can be recaptured. So Carlin uses this approach to generate complex barcodes by activating a Cas9 and targeting this barcoding region downstream of an EGFP transgene. This can also be recaptured. I'm sorry, my light's turned off. This can also be recaptured with mRNA capture methods. And I will be introducing a new method called mutable cell tagging that combines these methods. So mutable cell tags utilize two lentiviruses to confer the necessary components of this dual-channel lineage recording system: a Cas-guided cytidine deaminase to create edits, and a cell tag that contains a targeted editing region, or TER. The editor targets this dual protospacer area, enabling C to T transitions to occur within the TER. This occurs within a sequence less than 90 base pairs, which is generally compatible with short-read sequencing. And so this is actually really great because, we can use Ultima's platform and still get all of the information we need for this without having to adjust anything for read length. So because we have this this Cas-guided base editor, multiple edits are able to occur as these are non-destructive to the PAM motif. So we can effectively retarget this area of the genome. And when this method was tested with bulk RNA sequencing, we were able to capture editing rates of up to 25% for a single base and confirm that the adjacent cell tag was shielded from base editor activity. So I'd like to highlight that this is an improvement upon the existing technology. And from one intervention, we can create a different lineage tracing outcome than we could before. So with a method like cell tagging, we clonally label cells and find clonal relationships between cells in a population, categorized over here on the left. And with mutable cell tagging, we can create this high-resolution lineage tree within a single clone, showing how closely related single cells are within one clone. So we can actually create this lineage tree structure with this technology, as opposed to just showing groups of cells that are clonally related. So in order to create these high-resolution lineage trees, we would start with an initial cell population and transduce these cells with our base editor and cell tag TER lentiviruses. So in this case, I'm showing one cell from the starting population that has received 4 individual cell tag TER constructs by the 4 little lentiviruses shown here. These cells grow and divide and write lineage information on the TERs as they do so, and all cells derived from the cell that was tagged are in a clonal population. So all of these cells are within this one clone. And then we leveraged the PIP-seq platform from Fluent Biosciences in order to capture single cell transcriptomes and our lineage information that we're able to recapture with poly-A capture techniques, which is what Fluent uses for their mRNA capture in PIP-seq. So once we then proceed to sequencing with Ultima Genomics UG100, we can then use the sequencing data to form these lineage trees with computational methods such as Cassiopeia from the Weissman lab in order to resolve all these lineage relationships. So in this particular toy example, I want to kind of highlight how the technology works. So if we have one cell, we can detect both the cell tag and the TER that are affiliated with it. And we can have the same cell tag across multiple cells within the clone, but they can have different edits on that targeted editing region highlighted here. Since we inserted 4 cell tag TERs at a multiplicity of infection of 4, we actually have multiple cell tag barcodes that we can then relate to individual edits in this targeted editing region. And then the relationships between cells can be elucidated between the patterns that arise from these edits that are affiliated with these individual cell tags. So this allows us to multiplex this lineage information at quite a high level. And then using bioinformatic techniques, we can transform that sequencing data into a cell by character matrix to represent the edit information and run phylogenetic tree reconstruction algorithms with tools such as Cassiopeia to determine the most likely tree structure. So to illustrate the utility of these mutable cell tags and interrogate what drives successful cell reprogramming outcomes, we applied this technology to a multiple cell reprogramming paradigm. So starting with primary MEFs, which are considered the workhorse in the reprogramming field, we convert the components of the mutable cell tagging system. We then let the cells multiply for about 4 cell divisions, increasing their total clonal population as they do so from 1 cell to about 16 cells per clone. And then we split these cells into 4 groups after writing all of their lineage information in this initial clonal expansion phase. So when we split them, the first set, while they're still MEFs, we took as a state sample with PIP-seq, and the other 3 entered different reprogramming protocols. So we've decided on one reprogramming protocol that can represent each germ layer. So the induced endoderm progenitor, or IEP, the induced muscle progenitor cell, or IMPC, and the induced neural stem cell, or INSC. And each of these protocols have different reprogramming methodologies and kind of philosophies behind them. So the IEP for the endoderm has a 2 transcription factor cocktail and relies on constitutive expression of the HNF4 alpha and FOXA1 transgenes, while the MyoD1 reprogramming for the IMPC uses a 4-day induction and the master regulator transcription factor, MyoD1. And then for the induced neural stem cells, we actually use an OSKM pluripotency transcription factor cocktail with a 5-day induction and then direct their differentiation with neural stem cell media. So we wanted to have a large breadth and variety of these types of cell reprogrammings. So once the MEFs are reprogrammed into their final fates, we then take our last sample with PIP-seq, which we anticipated to be over 1 million cells at the beginning. I will say towards the end, we didn't have quite that many. We think we got about 700,000. So we were close, but we still needed that 1 million cell kit in order to get this done. So we wanted to find these really expansive clones. And with our starting cell population, even being relatively modest of about 30,000 cells, we had to have the scale in order to be able to answer this question, what are these clones doing? And so we used Fluent Bioscience's T100 and T1000 kits for 100,000 cell capture and 1 million cell capture in order to do this. And because of the size of this, Ultima Genomics made the most sense to be the sequencing platform we used for this. And because of the vast number of cells we have here, I'll actually only be showing a subset of this data today. So all the plots I'm showing you will be from about 100,000 cells, and actually only from a subset of the sequencing data, since we're actually still working on getting through it all, since we didn't realize, we didn't even have all of the processing power we needed for this scale within our own computing clusters. So yeah, definitely it's time for us to switch to the cloud here at WashU. So to start with, looking at our fate sample, we collected this with the T1000 PIP-seq kit and recaptured each of our anticipated final reprogrammed cell types, which we can highlight with the module scores here. And so we did see each of our kind of anticipated ending cell types. And then further, we can find these to break down into general cell types. So we, at the top of the UMAP, show that we have our IEPs. The kind of purple section in the middle is our neural cell types, and then the bottom kind of foot in blue is the IMPCs. And with Panglau, we were able to look into these cluster identities and see what all kinds of cells we were finding. So we can actually break this up into different types of IEPs. So we actually have quite a bit of heterogeneity in our final types, and that seems to have some type of mesenchymal to epithelial spectrum within these cells themselves. We also get several types of neural type cells from the INSC reprogramming. And then lastly, the IMPCs actually have an interesting overlap with the neural fates, because which is actually noted to be an off-target effect of MyoD1, that it sporadically binds with neural progenitor transcription factors. And so here I want to start looking into the lineage information, and we'll step through this data one by one with this clone, and then kind of all at once with the following ones, since the data will all look kind of the same. So here we have an expansive sublineage within one clone. So this piece of the tree was very expansive. And if we check the cell types of each of these cells related to the tree, they mostly are IMPCs and some INSCs, so the muscle and neural types that we reprogrammed. We can also assist gene expression to this tree, and here I've just highlighted some of the module scores, which I will admit look a little messy, but that has more to do with the refinement of these cell types. As we've said, we hope for good reprogrammed types, but they are tricky to particularly identify, especially when there's overlap. So from there, I wanted to kind of show you where these clones sat on our UMAP. So we are seeing them specifically in the IMPC and INSC regions. And the clone pattern here indicates that this subpopulation of the MEF clone favored the IMPC reprogramming protocol overall. And so that's different than this next clone that I'm going to show here, where it was actually so expansive, I can only show you a tiny bit of its lineage tree for being able to actually see all the branches here. So generally, this one reprogrammed into IEP cell types, so it does sporadically have other cell types arise as well. And so here we're only showing 100 cells out of over 2,000, but we can see that there are actually many types of IEPs that are generated from this, and that they're closely related in the branching structure. So this could indicate that these cell types are maintained within IEP culture and are not distinctly different fates. So finally, I have one last clone to share with you, and this is of course the most exciting one. So this clone contains several expansive sublineages with different reprogramming cell fate outcomes. So while this clone favors the ApoA1 high IEP outcomes, so this kind of top section here, in the bottom, these extra branches that are more distantly related actually seem to favor the IMPC cell fate as opposed to the IEP fate. And so in this clone, this seems to indicate that the IMPC cells and the IEP cells don't seem to reprogram from necessarily the same cell type since they're not closely related. And what's interesting and kind of patterns back to the other clones we were looking at, the INSCs are much more closely related to the other two outcomes. So it almost seems like the neural stem cells are able to be produced by maybe a generic reprogramming cell state as opposed to a more specific one, which may be for the IMPCs and the IEPs. So to take us back a little bit out of the fate cells, we're going to talk about the original MEFs that these cells reprogram from. So if we assess the state sample to understand that MEF population that gave rise to these reprogrammed clones, we find that most reprogrammed clones arise from clusters 11, 5, and 6. And from cluster-specific markers, we're able to see suggestions of neural crest progenitors in clusters 0 and 13, proliferation markers in clusters 5 and 6, and fibroblasts from other types of embryonic structures. And generally speaking, the cells that successfully reprogrammed in our system were from MEFs that were proliferative, which is generally supported by the field, in that highly proliferative cells are more likely to reprogram across many types of paradigms. So in conclusion, we effectively use the PIP-seq T1000 million cell capture kit and Ultima Genomics Sequencing to characterize our cell fates at scale. And as far as the quality of the data goes, we've really seen that this recapitulates other single cell assays and sequencing that we've done with 10x and Illumina sequencing that we have available here on campus, kind of in our core facilities as well. So we've seen all these same marker genes and been really, really pleased with the consistency we've seen in our data output. We've also found evidence to indicate broad reprogramming potential is present in cells, but sublineage fate restriction could indicate preferential cell states in specific reprogramming outcomes. We also find that those proliferative MEFs give rise to our final fates. And in the future, we're working to quantify lineage outcomes and patterns in a more systematic way. So figuring out how to kind of show these in a more many trees that have similar structures, sort of summary statistics. And further, we would like to apply mutable cell tags to more model systems as they can make high resolution lineage recording possible in 3D cell model systems, cell transplant experiments, and many other types of development and disease modeling. And so with that, I'd just like to thank the members of the Morris Lab, specifically Snow Yang and Naoto Yamaguchi, who helped a lot with these experiments and really played a critical role in getting this done. And I'd like to thank the teams from Ultima Genomics and Fluent Biosciences for their support with this project as well. And lastly, thanks to the institution, WashU, and our funding sources for the project.
Thank you very much. This was really great. We'll take the questions in the end, so I will take over the sharing and share the second presentation.
Do I need to stop sharing? Okay.
The answer is no. Okay. So today I will share with you how Ultima Genomics has come to really revolutionize the fields of single cell and how we can look at the future and what we can do with much more cheap sequencing with single cell analysis. So before we jump into the single cell part of the talk, I will first recap a bit more information about the Ultima Genomics and the UG100 machine. So about 2.5 years ago, we came out of stealth and told the world that we are ready to do the $100 per genome. And in this year AGBT, we actually did our commercial launch and announced that we're offering this machine and the UG100 and $100 per genome. A single wafer, I will soon talk about is a wafer, a single wafer, which is a single run, is $2,000. The UG100 is completely automated and can run 24/7. And in last AGBT, we showed that our indel rate, the F1, is above 99%. And I don't know if you saw our last webinar on PPM-seq. I encourage you to go see it. It's really a great webinar. And we showed that with the PPM-seq mode, which is a special sequencing mode that we can do, we can get to read quality of 60/70 with really 1 part per million, 1 error per million. And this is really very high accuracy and allows you to look at the rare SNPs. And as I said, 24/7 automation, which allows you to get to up to 20,000 genomes per year. So a bit about our technology. So here, this is the wafer. The wafer is a simple piece of a silica wafer. It's a very standard substrate in the semiconductor industry. And this is what we use as our substrate to sequence on. So we are putting our clonal beads on the wafer, and this is where we're doing our sequencing. The way we dispense our reagent is by simply dispensing them on the wafer and doing spin coating. So we don't need any sophisticated fluidics. We simply put our reagents and spin the wafer and coat it. It's very fast and very efficient. Thanks to such fast and fast imaging, we're able to get to 14 hours per run for 300 base per genome. So this is about the wafer. And a few words are about our chemistry. So unlike many of the other companies, we are not doing terminated nucleotides, meaning we are not flowing all 4 bases and looking for different fluorescent colors to detect which base was added. We're actually using flow chemistry, meaning we're flowing a T followed by a G, followed by a C, and followed by an A. And when you are flowing, for example, for this example, you're flowing a G, and you have 3 C's in a row, so all G's will enter together. And because we're using non-terminating chemistry, we can actually use mostly natural nucleotides because we don't need a terminator in each of them. So most of the nucleotides that we are adding in our run are natural, and only a percentage of them have fluorophores on it. And this allows us to get to much longer reads in higher quality. So currently we're doing 300 bases in very high quality. And also we're using 300-fold less polymerase, which again reduces the price. So this was about the machine. So what about the single cell? So really there is a revolution in single cell field that is unfolding. So if we go back to the future only 2-3 years ago, so 2021, if you wanted to do a 5,000 cell experiment, your single cell RNA library prep would have cost you about 10 to 20 cents per cell. And your sequencing reagent cost would have been, again, also 5 to 10 cents per cell. That would put your cost per sample for the 5,000 cells that you would want at about $750. But over these last 2-3 years, there was really a revolution. Both the sequencing price has dropped down, and different companies, starting from 10x with new products, and different companies that enter the field like Fluent, Parse, and Scale, have really dropped the price, not only in sequencing, but also in the library prep. So when you actually want to do single cell at scale today, you can actually get to a price of about 1 cent per cell, and even lower than 1 cent per sequencing. So that really puts your cost per sample at about $100, opening really new fields, opening clinical diagnostic, and many, many other things. So what is the space of large-scale single-cell applications? So the most obvious one, which already exists and needs to be expanded even more, are atlases. So there are huge atlases out there already. This is a collection of atlases that has about 100 million cells. There are atlases of 20, 30 million cells. Many of those atlases are really great. They should be expanded with maybe more cell types, maybe more samples from different individuals. Many of them are very, very heterogeneous, meaning they have either many different library prep being made, many different labs have contributed to them. So I would argue that there is also a need for big atlases that will be more homogeneous, which, because many of those atlases will actually be an amazing data source for many of the AI possibilities that we have in the future, and I will talk a bit about it. Another field that is part of the atlases is the spatial atlases. So they started to come out. They're still very small, and they will probably grow in the future. And again, in the last year, we have seen 2 companies come out with amazing spatial transcriptomics products. We've seen 10x that already had the Visium, which was a great product, and they made it even better with the Visium HD that gives you a 2-micron resolution. And also we have seen Curio come out with two product, Seeker and Tracker, which really allows you to do spatial transcriptomic at scale, and again, contribute to huge atlases that I'm sure will come in the near future. One of the biggest fields of large-scale genomics is the perturb-seq. So in 2016, there came 3 papers by Jonathan Weissman, Aviv Regev, and I think one by Amit. And what they have shown is that you can take a group of cells, millions of cells, and you can do a sgRNA library, a CRISPR RNA library. The sgRNA will randomly enter different cells and perturb your gene of interest. And then you can do a single cell experiment and look at all the perturbation at once on different cell on the different cells that got the perturbations. And the amazing thing about this experiment is that there are really no limits. You are not waiting for samples. You can just take your cell type, grow it to a billion cell, take your guide RNA library, order 100,000 different guide RNAs, and make this experiment as big as you would like. And really, we have started seeing such large-scale experiments coming out, and more of them are needed in order, again, to build the perfect data for the possibility that we have today, the possibility to take really AI, machine learning, and take this data and come out with understandings that we are currently not able to. And just a day ago, even I think 2 days ago, came out a beautiful paper termed single cell GPT. So they have taken the name of the generative model. And what they showed, they're taking a huge atlas, 33 million cells, and they're training on it with a generative model. And their claim, which is a claim that I would buy, is that as you look at natural language models, that where you have letters that make words and make books, you also have genes that lead to expression and leads to the cell types. So in the same way, you can just feed the model with a lot of data and start learning new things from the data that really we are currently limited with what we can learn from such huge data sets. And in here, they used an atlas of healthy tissue, an atlas of normal cells. We can only imagine what can be done with this when you start adding in the perturbations, all million cell perturbations that will really show the generative models what the language is. Another field which is currently really only emerging is the field of single cell sequencing in clinical application in cancer. And again, I don't have a lot to say about this today, but I'm sure that in a few years, a lot of new things will happen in this, again, because the huge price drop that brings the cost of a sample to about $100, you can start thinking about really doing diagnostics in this field. And the last thing that I'm going to talk about in the large scale things that can be done is actually not single cell RNA. We're not limited to single cell RNA. We can, from a single cell, we can take the genome, we can take we can look at methylation. So this year, the NIH started the SMAT program, the somatic mosaicism, where they started looking at different heterogeneity in our body. So when we're doing whole genome sequencing today, we're usually taking a saliva sample or a blood sample, and then we're deciding this is the genome of that person. But actually, if you will look at different tissues, if you will start looking even within a tissue, you will see a lot of heterogeneity and many, many different SNPs that arise even in single cells within the tissue. And I will show a bit of more data about this on this later. So I talked a bit about what we can do in single cell, like what are the big applications that we can look at. And now, how do we actually do single cell at scale on the UG100 machine? So as I said in the beginning, the machine has a 24/7 automation. The price per million read, the sequencing reagent cost per million reads is 30 cents, and a single wafer will cost $2,000. If you do the calculation, you will see that on a single wafer, we can do about 250,000 cells per run. This brings the cost of a single cell to below a cent of sequencing. We can do about 5 million cells per week, which gives us about 200 million cells per year, more than 200 million cells per year on a single UG100 machine. So how do we do single cell on the UG100? So the first steps are exactly the same as you would do today. Nothing has changed. Like if you take your blood sample, if you take your tissue sample, you simply dissociate the cells, you collect the cells from the blood sample, and you go into your favorite single cell RNA library prep, whether it will be 10x, Fluent, Parse, or Scale, and you do the library prep as you would before. After we have done with the library prep, we do a simple conversion into the UG adapters, which I will show a few more details about it in a second. And then we go into the sequencing on the UG100. So in order to show how we are doing our conversion, I will take something that is probably known to almost everyone in the chat, the 10x 3' single cell RNA library prep, the most widely used single cell library prep. So in the normal 10x, we have the gems, the emulsification, where you have a bead and a cell together. Then you do the cell lysis to the cells, and you do your RT reaction, getting your cDNA connected to the bead barcode. And then in bulk, you do your cDNA amplification, cleanup, and you actually do the library prep. And you end up with a read that looks like that. So we have P5 on the left side, followed by read 1, the 10x barcode, which is the cell barcode, the UMI, and a poly-T. And on the right side, we have the P7 and the sample index. So when we want to take this structure and convert it into the Ultima structure, what we do is we do a simple PCR. And because Ultima Genomics is currently doing single-end sequencing, we want to take this sample barcode and actually switch it to the other side. So what we do, we are doing our PCR with a UG primer A that will have an index that will actually swap the sample index that is found on the I7 side. And then after a few cycles of PCR, we get this kind of structure, again, the same structure, only with our primers and our own sample index. And then we do our ePCR to create our clonal amplification on the beads, and we get our beads ready for sequencing. When we're doing our sequencing, we are starting from the UG primer A site, and we're going inside to the bead, and we're sequencing through the index, through the R1, and through the poly-A. So what we're seeing here is actually the signal intensity that comes out of a run. We have here 116 cycle, which is each cycle consists of 4 flows. As I said, we are a flow chemistry company. We are flowing a T followed by a G followed by a C followed by an A, 116 times in this run. And you can see the signal intensity for the different nucleotides, for T, G, C. And what we can see is that in the beginning of the run, everything is very, very peaky. That is because we are going through the barcode and the constant R1 region. When we're getting to the cell barcodes and the UMI, things become much more homogeneous. And then we go into the poly-T region, and we see a huge spike in intensity because we are reading through the poly-A using T's. So actually reading through the poly-T is a bit of problematic. So I'm going to show you why in this mock example. So we have here a UG bead, one of the 7 billion, 8 billion beads that exist on the wafer. And as I said, we are a flow chemistry, so we're flowing a T followed by a G. So in this mock example, we have the CAT as the primer, and the next nucleotide to be entered into the bead is a C. So we are now flowing a C. For this particular bead, nothing will happen. Then we flow a G, again, nothing will happen. Once we flow a C, the polymerase will extend the reads, and we will get a signal. So then we continue, we flow an A, again, added, and then we go to the next cycle. So we finish the single cycle, and we go to the next flow, which is, again, a T. In a regular situation, if we just flow a T, most of the beads on the wafer will get to the poly-T region, poly-A region at the same time, which means there will be a huge nucleotide depletion. There will just be not enough T's to cover all the beads on the wafer. So the way we solve it is by doing poly-T polishing, poly-A polishing. Again, the poly-A and the poly-T are interchangeable. And we're just giving a lot of extra injections of T just to have enough T nucleotide to go through these long regions of poly-A, which is basically the poly-A primer. It's 25 bases coming from the 10x design. And then we are passing through it and are able to sequence the cDNA that comes after that. And actually, when we do that, there is actually even a small advantage for us, because what we sequence is exactly the 3' polyadenylation site of the molecule. We're not sequencing a fragment molecule, the 5' side of a fragmented molecule, but we're actually sequencing exactly the 3' side. And I will not go into the importance of alternative polyadenylation. There are a lot of isoforms that use alternative polyadenylation, but I will just say that already there are tools out there that will take your single cell RNA data and look for alternative polyadenylation sites using our technology, which is very accurate on the location of the 3' polyadenylation site. So this was the sequencing. So we are done with the read. Now we actually have data. So what do we do with it? Again, because we're a single-end read company, the way the reads look like is in a way that most data processing tools will just not know what to do with. So we actually have on-tool data processing that takes this read structure, trim and discards the things that we don't care about, like the polity and the read one, and takes the cell barcode, the UMI, and the cDNA, and actually simulates paired-end, not paired, paired-end FASTQ files. And those paired-end FASTQ files will be able to go directly into your favorite data processing tool, whether it be Cell Ranger, ParseBio, StarSolo, whatever you would like. It will just fit into it immediately without no changes. Okay, so we talked about how we do the sequencing. So now let's show some data that came from the UG100 on the single cells. So we'll start with the big, large-scale perturb-seq. So this is a paper, again, from the Weissman lab, coming 2 years ago. And in this paper, they have taken 10,000 different sgRNA guides and used 2.5 million cells in order to find the different perturbations that can be done with it. And in this paper, they actually did both Illumina sequencing for the 2.5 million cell and Ultima sequencing. So what they show here is that there are very high correlation. This is actually looking directly at the perturbation, not the single cell expression, but on the differential expression. And they can see that there is very, very high correlation between Illumina and Ultima. And just this year, I think, yeah, it came out this year, there is a great paper coming from the Rhodes Satija lab, a work by Longda, Carroll, and Appy. And in that paper, they looked at 6 different cell lines. So unlike the earlier paper, which took one cell line and just perturbed many, many genes, here they took 6 different cell lines and actually focused on 5 particular pathways. They focused on interferon gamma, interferon beta, TGF beta, TNF alpha, and the INS pathway. And they perturbated 271 genes in total, and 2.5 million cells were profiled. And by using different cell types, you can actually look at how different cells react to the different perturbations. So I'm just going to show one small vignette from this paper. So looking at the interferon gamma pathway. So we have the core components of the interferon gamma, the actual receptor and the actual pathway. So we have the interferon gamma and the JAK1, JAK2. And when we're looking at the perturbations done on the core components, we can see many, many genes that react more or less the same between the different cell lines, and they go up and down depending on the perturbation. And the sensitivity of the perturb-seq is actually great, that you can also look at the secondary response and primary response that comes from the transcription factors that are induced by this receptor. So when we're going for transcription factors or transcription repressors, you can also see a very nice reaction to the perturbation that again goes in more or less the same manner between the different cells. And again, this is just a small vignette. I encourage you to go to the paper and see. Again, beautiful data and large-scale perturbation. We have also done some comparisons between Ultima and Illumina. So this is a paper that came last year from Simonson and I from the lab of Joshua Levine. And here we compared different blood cells. And what you can see, first of all, these are UMaps that just show that both systems capture all the cell types in the same manner. And you can look also at the proportion of cells and see that there are no observed differences between Ultima and Illumina, very small differences. For the parse bio WT, I actually just showed you the data. The beautiful paper by Raul Satija was all done using parse bio WT kit and was sequenced on the Ultima platform. We have also done a comparison on the 10x Flex, which 10x Flex is actually really nice to sequence because you're only sequencing a small barcode, like a 50 base that of hybridization site, which is the a kind of a barcode. You can look at it. And first of all, it's very, very short. We can do even more runs per week. And you can see that the correlation between Ultima and Illumina is really, really high. And we also look at Fluent. I don't think I need to convince you that Fluent data looks the same. I think Sadie have done it perfectly. Okay, and the last example that I'm going to show is on actual single cell whole genome sequencing. So this is done together with Bioscribe. So Bioscribe has developed a kit that is based on a technology called PTA, Primary Template Directed Amplification, which actually gives you an almost perfect amplification from a single cell. You can actually get the entire genome with an even coverage by just randomly capturing different parts of the genome and get a very, very even coverage across the genome from a single cell. And that allows you to do even 15x or 30x and find different single nucleotide variation and structural variations. So in a work together with Dan Lando, with the students Dennis, Tamara, Ivan, Clayton, and John, we have gone and tried to look at 1,000 cells with using single cell whole genome sequencing. And Dan Lando has always wanted to do those kinds of experiments, but when he wanted to do them 2 years ago, he had a problem because, first of all, the production was very labor intensive. It took 12 weeks. Analysis was crazy intensive. And the sequencing cost was really not affordable. You couldn't go and do this experiment. 2 years have passed. Lando Lab have perfected the production stage and the analysis stage. But thanks to Ultima Genomics, we now have also a sequencing cost that they can afford and go forward with such an experiment of doing 1,000 single cell. So again, these are vignettes. The first what they wanted to see is, does it actually work? Can you take single cells and build a nice lineage tree going back to the first single cell in this clonal compartment? And they took the DLD1 cell line and sequenced using Ultima to 15x coverage. And what you can see is that they can really build a beautiful clonal tree going back to really a single cell. Secondly, they actually took aging people ranging from the age of 75 to, I think, 85, and they look at the single cells coming from the esophagus. And what they can see is really that in each single cell, they can find hundreds to thousands of different single nucleotide variation, and they can also find really clonal behavior, where you can see a P53 clone that really has the two types of P53 mutations, and the clone just exists there. It just lives there and is happy, and you can find it in the single cell level. So this is an amazing work, and I hope it will be published really soon. So to summarize, I will go back to this slide, which really says that we are in the process of really what I think is a revolution, something that just cost about $750 2 years ago, now can cost you $100, and you can really now do that in scale. The library prep from Fluent, from Parse, from Scale, and from 10x have really changed over the years, and now a 1 million cell experiment like Sadie has shown us is not something you will need special equipment to do. You can just do it in your own lab. So thank you, and we'll be happy to take your questions.
Thanks, guys. Super informative. I have a few questions that are sort of coming in right now. These first two questions are for Sadie. What part of the analysis was computationally intensive, the gene expression analysis or the lineage reconstruction?
Yeah, so I actually would say neither. It's actually been more of an issue on the alignment for us because we're just hitting memory issues with our high throughput computing cluster that's housed here at WashU. So Some of the issues we're running into, like we know we would not run into it if we actually just had done this on the cloud, but that was something we weren't set up to do. This is the first time we're actually working with data this large with this many reads, and trying to create a BAM file this big has been just challenging for us. So working with our kind of data administrators and trying to figure out how to do this on our system has been what has been challenging. Actually running, after getting that kind of done, running the gene expression analysis and working on the lineage reconstruction has been really streamlined. So that's actually been pretty quick to do.
Great. And then the second part of that question was, within the proliferative MEFs, which were there subsets of cells that were more likely to give rise to the 3 separate differentiated cell types?
So transcriptionally, those groups did not look different. They all were present in each of the clusters. So of the ones that I highlighted, it was 5, 6, and 11. So that doesn't necessarily mean they're still the same cells. So we've seen in other lineage tracing work from the lab that if we kind of split the cells into 2 pools and look at clonal information across an ATAC assay and an RNA assay, that we can see differences in other modalities, such as in chromatin accessibility. So I would think that there are differences there, but I just think we're not capturing them yet, at least not transcriptionally.
Great. This is a question probably for Zohar. Does Ultima plan to create or partner to create alternative library prep kits or add-ons in order for researchers to directly make an Ultima library, so I guess maybe a native library, and reduce the worry of PCR bias?
Yeah, so first I will comment on the PCR bias. We are minimizing the PCR bias by doing as few cycles as possible. Like usually you have a lot of material coming from single-cell RNA library prep, so we'll do 4 or 5 PCR cycles in order to get our primers on. Secondly, yeah, the question is, of course, we are partnering and we're trying to get it. I don't know the timing that it will happen, but we are hoping to get native UG libraries on some of the vendors.
The next question is if Ultima sequencing is compatible with 10x, TCR, BCR, single-cell RNA-seq.
So I will just say what the problem is with sequencing TCR and BCR. It's just very, very long. The VDJ and the constant region that exist there are also almost 500, 600 bases in length. So this is one of the really nice places where per-den sequencing really helps, because you're just sequencing the VDJ region, which this is what interests you for one side, and the cell barcodes on the other side. So going with a single-end read, we will not reach the other side. We do have a library prep that uses circularization and allows us to get the both sides of the molecule. And we hope that in the near future, we'll have new ways of doing it in a very efficient way.
Okay. There's another question just asking when the webinar recording will be available online. It'll probably be available early next week. So we'll be sending out an email for anyone that registered, and also have it on our website. There's another question for Sadie, which was why Fluent over the other single cell providers like Scale, Parse, 10x?
Well, I got an email from Fluent saying, hey, we know you guys do single cell if you thought about us. So we'd actually started looking at Fluent, I think about a year ago, maybe more than a year ago. And so, we just looked at their papers, looked at their white paper, and, we thought it was really promising. And we thought that the scalability and being able to do it like so simply in one tube was really intriguing. That being said, we are now looking at Scale and Parse as options as well, because there's, a great use case for each of these different technologies. Like, we don't think we're going to, stop doing 10x necessarily. Like it's, something we've had a great experience with so far. So yeah, I'd say it's mostly just they had some really personable salespeople who convinced us to get started. So we did, and they had some of the most scalable kits out there that we could see ourselves using, in our current space. Like right now, I know that we are getting started, I think, with Scale, and there's just going to be, some different things to learn about it. And so, we took a look at that protocol and thought, this looks like we can do it. It looks pretty easy, and it was approachable.